Good morning, dear students. Welcome to Genesis. We have finished 4 chapters of our syllabus and this is the 5th chapter, Transpiration. I hope you are liking my videos. I would just request you all to subscribe and like my channel. That will be a great motivation for me. What is transpiration? Transpiration is that phenomena by which a plant can lose water in the form of water vapor from leaves and other aerial parts of the plant. Aerial parts, we refer to those parts which are above the soil. Those are known as aerial parts. We know a lot about transpiration, like how it helps the plant to cool down its body, how it generates a suction pool which helps the plant to draw water to greater heights. But in this chapter, we will try to prove the process of transpiration with help of few experiments. So let's quickly start with experiment number 1. In experiment number 1, we have taken a potted plant, a healthy potted plant, and we have covered the aerial part of the potted plant with a polythene bag, and we keep this setup out in the sun. Along with this setup, you are also going to keep a control. In the control, instead of a potted plant, what are we keeping? You can keep a stick or you cannot keep anything, just a support to keep the polythene bag intact and keep it out in the sun. After some time, you are going to observe appearance of water droplets on the surface of polythene bag in setup A. Whereas in setup B, we do not find any water droplet on the surface. What does it mean? It means that here, since the plant is present, it undergoes the process of transpiration and releases water vapor. This water vapor, when comes in contact with the polythene bag, undergoes condensation and gets converted into water droplet. Such a phenomena is not happening here because there is no plant. So the experiment 1 proves that during the process of transpiration, plants produce or release water in the form of water vapor. Now let's jump to experiment number 2. Experiment 2, we have taken 3 setups. In the first setup, we have named it as A. We are taking a potted plant, but make sure that you cover the pot along with the soil within a bag. Why are we doing this? We are making sure there is no loss of water from the surface of the soil by the process of evaporation. And we are going to do this for both setup A as well as setup B. So we cover the soil with a bag and we tie it tightly so that there is no loss of water by evaporation. In the first case, we just keep a potted plant, but in the second case, we keep a potted plant, we keep a plank of wood, and to that we have attached cobalt chloride paper. The third setup has no plant, but it has a plank of wood and to it is attached a cobalt chloride paper. If you remember, in 9th standard we studied that cobalt chloride paper is an indicator of presence of moisture. So otherwise in nature, the cobalt chloride paper, when there is no moisture, it is blue in color, but when it comes in contact with moisture, it immediately changes to pink color. In the first case, only the water droplets can be seen on the surface of the glass. But in the second case, we find that there are water droplets. Along with it, there is a change in the color of the cobalt chloride paper. Initially, it was blue in color and with time, it changes to pink, which clearly indicates that these are water droplets because cobalt chloride paper changes color only when it comes in contact with the moisture in the air. So it has changed from blue to pink in color, which is 100% confirmation that there are water droplets that were released by the plant during the process of transpiration. In the third case, there is no plant, there will be no transpiration, no water droplet, there will be no change in the color of the cobalt chloride. So it remains blue. So now let's go to the next part of the chapter where we are going to see how we can find out the amount of water lost during the process of transpiration. In this, I have drawn 3 experimental conditions which we are going to use and see if we can find out the volume of the water loss during the process of transpiration. Remember one thing, transpiration, it is that process by which a plant loses 98% of the total water absorbed. It only uses 2% of the total water for the process of photosynthesis. Now let us see, in the first experimental setup, what we have to do is we will take a potted plant and we will tie up the upper surface or the soil of the potted plant with a polythene bag just to ensure there is no loss of water by the process of evaporation. So before you keep it out in the sun, you will weigh it, and then after keeping it out in the sun for some time, we again weigh it back and we are going to find a decrease in weight. Why? Because the soil lost its water not by the process of evaporation, by the process of transpiration. So that is one method by which you can guarantee and find out the weight difference that happened due to the process of transpiration. Now if you want to find out the the volume of water that is lost by the process of transpiration, an approximate amount. Remember, whatever loss is happening, some of it will be taken up to perform the process of photosynthesis, and rest of it will be lost due to transpiration. So it will be an approximate amount. So what do you do is you take a glass jar, fill it up with water, put a potted plant, fill it up with water, put a plant inside it, and it's a rooted plant. Okay, what we do is, with the help of a rubber, we are going to attach a graduated tube to it. What is a graduated tube? A basic glass tube with marks on it. Okay, now you fill it up with water to such a level that whatever is the level of the water, you mark it. After some time, observe the level of water in this tube. You are going to see there is a dip in the level of water. You subtract the value, and that is the volume of water that is approximately lost during the process of transpiration. The third process is, you take a simple test tube, and you do not have to take a rooted plant here. You can just take a cut shoot of a plant and fill the beaker with water and cover the surface with oil. This is very, very important. Why are we using oil here? Because again, we do not want to lose any water by the process of evaporation. Just to avoid that, we cover the surface of the water with oil. Now keep the twig as it is and look at the amount of the water that is present here. After some time, when you again try to find out the level of the water, you're going to find a dip in the level of water. 100% it was not because of loss by evaporation. So this was lost by transpiration. So these are the three methods by which we can get an approximate value of the water lost by the process of transpiration. Now let us look into the next part of the chapter, which is about a very unique device known as potometer. Now potometer is a device that can measure the rate of water uptake by a plant, and this is equivalent to the amount of water lost by transpiration. If you remember, I have told it before also, plant loses almost 98% of water just by the process of transpiration. So whatever is the amount of water uptake by a plant is almost very equal to the amount which is lost by the process of transpiration. Now to find this rate, this device potometer got invented by many scientists. So there are various kinds of potometer. Darwin's potometer, there's Farmer's potometer, but the one which is in our syllabus is called Ganong's potometer. So this is how Ganong's potometer looks. It has a reservoir, it has a capillary tube. To one end of the capillary tube, we basically attach a twig of a plant. But while you are doing it, remember one thing, you have to seal the topmost level completely, because then only you make this entire system airtight, which is very essential for this device to work. Now this portion of the capillary tube is graduated, that means you can see there are markings on it, and the other end of the capillary tube is actually dipped in a beaker which contains water. So there is water even in the reservoir, there is water in the beaker, but there is a massive difference between the two. We have colored this water. So you can color the water by putting eosine or maybe potassium permanganate, anything that will give it a bright color, and it should be very easy for you to understand the movement of water through the capillary tube because it is colored. Now what do we do with this part? Now you first basically run the reservoir and you allow the water to wash the entire capillary tube. Once it is done, you use the stopcock and close the reservoir, pour water again. Now you have dipped one end of the capillary tube in the beaker. What we do is, we slightly, very slightly, we take out this capillary tube out of the level of the water and allow air bubbles to enter into the capillary tube. So once you do that, there are going to be many air bubbles which are going to enter into the capillary tube, but that's not what you desire. You want only one air bubble to enter into the capillary tube. How can we achieve that? Again, you open the stopcock, let some water flow into it. This water is going to push the excess of air bubbles that are there and immediately stop the reservoir once you get to see there is presence of only one air bubble in it. Now you take this setup or keep this setup in a place which is well lit, there is enough sunlight, and observe what's going to happen here. What will you observe is there will be movement of the air bubble along with the colored water into the capillary tube. Now what is causing this? Because once we started this, you cannot expect the colored water to just like that enter into the capillary tube. It enters into the capillary tube only when there is a suction pull that is generated. And what must have generated that suction pull? Loss of this plain water by the process of transpiration through the twig. So when the amount of this plain water started to reduce, there was a suction pull that was getting generated here, and that must have pulled the water, colored water, into the capillary tube. So what do? We observe the time when this colored water must have reached this part, so which is basically 0, and from there on you start recording the time. As this time proceeds, you're going to see that the colored water will reach from 0 to 1 or maybe to 2. Note down that time. That will give you the volume of the water that has moved up and the time that it has taken. So indirectly it is actually giving you the rate with which the water is being taken up by this twig. Why? Because the twig is taking this white water, which is causing the colored water to enter into the capillary tube. So they are almost equal. So this is giving you the rate with which this plant is uptaking water, and this is equivalent to the rate with which the plant is losing water by the process of transpiration. Ganong's potometer is an excellent device, but it has some limitations and some precautions also which you have to take before you set up this. So let's talk about the precautions first. Number one, this entire setup has to be airtight. So wherever you find little bit of openings left, you have to fill that opening with Vaseline or with anything that will make it airtight. Number one. Number two, the plant twig that you have taken, when you are cutting the plant twig, the cut has to be oblique. Why do we do that? Because it increases the surface area of the stem. So for example, if this is a stem and if you cut the stem straight, this is the surface area exposed. But if you cut the same stem and give it an oblique cut, it increases the surface area that is exposed. So you are going to cut it obliquely. And one more thing, when you cut this small twig, you have to do it underwater. You already know why it is, because in my last experiment I had told you that when you cut the twig underwater, you ensure that there is no air bubble which can enter into this twig, because if air bubbles enter into the twig, they are going to hinder the absorption of water by the twig. Okay, now let's discuss the limitations of Ganong's potometer. Entering air bubble into the capillary tube is not an easy task. It's quite difficult. Second, the plant twig, it may not remain fresh throughout the experiment. And number three, the air which is around this experimental setup, if there are changes in the air during the experiment, then it may affect the experimental results. Like, how do I explain this? If I keep this setup in a place which is very sunny, it will give you a different kind of result. If you keep it in shade, it's going to give you different result. And if you keep it in a place which is very windy or probably there is a fan nearby which is running, that also will give you a different result. So while you are doing this experiment, you have to make sure that the conditions are maintained throughout the experiment. Okay, there cannot be slightest difference in the the conditions that are there in the experimental setup. So with this, I finish today's lecture. I'm going to meet you soon in my next lecture.